So next up, I'm very excited to introduce Beatrice Demarchi from the University of Turin, who will talk to us about shotgun proteomics. And according to the title, they are not quite bulletproof, so this will be a very exciting talk. Go ahead.
Thank you so much, Zandra, and everybody in the community. It's really, it's an honor to be asked to give a talk on shotgun proteomics. I guess shotgun paleoproteomics, really. Going back to what Sam was saying, I was there when it was invented, so I'm that old. But I think it was actually quite a challenging talk to put together, and I've decided to go for a basic principles type of approach, because there is so much work that has been done in the last 15 years that it was sort of impossible to cover everything, improvements in technologies from, applications, which of course we will hear from about later. So as I usually do, I wanted to start with archaeology. I will also finish with archaeology real quick. So I guess as Sam was saying as well, we need to put ourselves in the shoes or in the minds of archaeologists first and foremost. Here we have a mildly famous archaeologist, Gordon Childe. Now, I think Gordon Childe must have been worrying about pots all the time. He must have been thinking about pots. He must have been dreaming about pots. He must have been thinking, what on earth were people putting inside these pots that, we find pots all the time, but what's in them? Now, unfortunately, he couldn't really reach out to Jessie Hendy, for example, and her group, who've spearheaded a lot of the work on shotgun proteomics in pots. But I think this sort of highlights the temporal and somewhat philosophical lag that we have in our field, where we are propelled forward, and we are ourselves sometimes propelling forward a field that is young, is dynamic, and is I think Matthew's going to talk about this later, is also leaning on the shoulders, using a lot of the technological improvements that biology is doing, but always going back to more ancient and fundamental questions in the way we approach our scientific inquiry. So real brief introduction. So this is the range of samples that can be used for shotgun proteomics. This is in the last 10, 15 years. We've moved from the first, I didn't put the reference, but everybody knows the first mammoth proteome publication by Enrico Cappellini and team. And we moved to encompass a really wide range of samples. And so we can recover proteomes and from this complicated and varied set of samples in a way that was not possible a short time ago. But the key thing is that we have now moved from a really sort of point, approach to large-scale studies where we can answer some really big questions about, I don't know, cultural heritage, some more cultural historical questions, or the phylogenesis, the evolution and the origin of certain organisms. Or my Zoom just died.
Yeah, I am not sure what happened there. Let's try again.
Can you still see the screen?
No, we can't see your screen anymore.
No.
There we go.
All right. No idea what happened there. So I was saying that we need to access the set of proteins that is expressed by an organism, and this means that we will have many hundreds of different amino acid sequences. Each of these sequences will have certain modifications, either post-translation modifications or diagenetic sort of changes. So we have a really complex starting point. But with these shotgun approaches, we sort of lull ourselves, I guess, in thinking that the analytical techniques that we use are actually so powerful that we don't need to worry too much about the starting complexity of this proteome. As I said, this may or may not be the correct thought, the correct approach. But the point is that really our lab protocols, our sample prep is not that complicated. They're fairly standard. The point is to get to a point where we have, as you can see here, basically a digest or a fairly clean set of peptides, and these we generate with diagenesis, with digestion or by diagenesis. So we know that our proteins are so broken down that we don't even bother digesting, for example. And then after that, we use a combination of separation techniques. We have high pressure liquid chromatography, typically HPLC and tandem mass spectrometry, and we generate thousands of mass spectra. And so from these, we can reconstruct the type of proteins that we have in our sample and then do more complicated things. So for example, we can compare proteomes across different sets of samples. And this, of course, means that we need some really state-of-the-art type of instruments on the one hand, but also really sophisticated bioinformatic approaches as well, because we need to handle a massive amount of data. And we need to do that within reasonable timeframes as well. So how does this actually work? I thought that me being Italian, I could use an analogy, and it's not cultural appropriation. I'm Italian, so it's okay. So I'm going to use pasta mista as an analogy. So pasta mista is something that we quite like to put in soups. So as you can see, it's a mix of different shapes of pasta. Is also, if you want, is the bits that remain after the production of the main, types, shapes of pasta. You put them together, and then it's quite nice to put them in soups. So let's imagine that we need to characterize the pasta mista proteome. So, as I said, we have a complex mix, so we need to separate it into its main components. And we said that HPLC, high pressure liquid chromatography, can help us do that. And it does that by exploiting the different affinities of the different analytes for a stationary phase, also called column, or a mix of solvent, the mobile phase. And we usually use a reverse phase chromatography. So the stationary phase is hydrophobic and the solvents are hydrophilic, but they're used in a gradient as well. But the point is that if we sort of force an initial mixture of compounds to go through this column, they will be separated out because some of them will stay in the column a little bit longer and some of them will go a little bit more quickly. So let's go back to our pasta. So if we use pasta as an analogy, I've run thousands of pasta shapes as standards, and I know that my farfalle usually come out around 7 minutes. So their retention time, they spend 7 minutes in the column and then they come out. And so if I've done that for all my compounds, then I can sort of characterize the components of my mixture or separate my analytes. However, if we think about pasta mista, it does contain a few of these components, but mainly it contains, well, it contains thousands of compounds, and quite a lot of these will also be all broken up quite badly because they get squashed in the shop or whatever. And so we can't really use this approach. We need something better, and this is where mass spec is the way to do it. The technical problem was that you can't just take a liquid and bash it into a mass spectrometer. That is not a good idea. Don't try it in your lab because people will not be happy. So very, very clever people came up with a range of soft ionization techniques. Sam has talked about Zooms. Zooms is based on MALDI, a matrix-assisted soft ionization technique. And here we typically use electrospray ionization. And electrospray ionization basically uses an electric field and a nozzle basically to turn a big drop of liquid with peptides inside into an aerosol of ionized peptide. And this means that we can transfer now peptides from the liquid chromatography type of system into the mass spec without breaking anything. So in a typical tandem mass spec experiment, we use a mass analyzer. Here I put a quadrupole, for example, and we produce a first spectrum, MS1. And from this, we can select. Typically, we tell the instrument the things that we're interested in, or we say, just choose the most abundant peaks or whatever. And we say, just go and try and figure out what these peaks are. And to do that, we need to fragment them. For example, here I've highlighted this 483.77 precursor. And what the instrument is able to do is to induce fragmentation of this peptide with that specific M over Z value. So it's first accelerated. So we apply again an electric potential, so increase the kinetic energy, and then we make it collide with neutral gas molecules such as helium, nitrogen, or argon. And in this collision, some of the kinetic energy is converted into internal energy. Basically, it breaks up into different pieces. Now, conveniently, it tends to break between, like the peptide bond is the first thing that tends to break. And what we get is a MS2, is a product ion spectrum, and we get thousands of these product ion spectra, and these are the ones that we can use to sort of figure out what's in our sample. So more specifically, we get B and Y ions when it's the peptide bond that breaks, and then we get A, C, Z, X ions when there are other bits that basically break off. And then we also get neutral losses of water, ammonia, and so forth. So if you want, you can try and follow what we're going to do with one of these peptides and scan this code, unless you know amino acid residue masses by heart, which I'm pretty sure many of you are, well, do. Sadly, I do. Some of them, not all of them. So if you've done that, we can now try and do some de novo sequencing together. We can do this by hand, and there are several tutorials that explain how to do it. I've put one here. Basically, don't think about it too much. Just follow the rules is my, there are rules, just follow them. Sometimes it's a good idea not to think too much about stuff. So we start by trying to find the B ions. The B ions are the ones that derive from the peptide bond breaking and the charge staying on the N terminus. So that means that we can see them. So we have the first rule that we use the neutral mass and we remove 17, the OH group, and that gives us a first number, which is 948.525 in this case. And what we do with it is we imagine it somewhere around here. And then we say, oh, what's the first big peak that I get at this end of the spectrum? Well, it's 819.487. Let's figure out if when I take 819 blah, 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 off 948, I find the mass of an amino acid, okay? So this is really a bit of like Sudoku. And luckily for us, we immediately find a mass that fits. We find glutamic acid here. So of course, it could have been this peak or many of the other peaks that we have in the spectrum. This is a simplified example. But if we keep going, then I've highlighted already in blue the B series, and we can go back until we find the whole B ion series. We can confirm this by looking at A ions, for example, or ammonia and water losses, which we find usually associated with B ions. Much better, well, on top of that, we also need to find the other sequence, which is that of the Y ions. So every time we find a possible B ion, we also calculate the Y ion. Again, just follow the formula. We apply this formula and we start by finding the first Y ion. And then again, we try and fit the masses of the amino acid between peaks, okay? And so there we go, we can find the whole series here because this is an example. In reality, this doesn't happen, okay? But we cross-check that we have the same sequence going backwards and forwards. So B ions go forward, Y ion go backwards. We put them together and we get farfalle, all right? So we got a peptide and it's the sequence and this was the precursor. As I said, of course, this is not what happens in reality. We have many more peaks. Most of them won't be assigned. Quite often you get, your series gets, it stops because not everything gets fragmented, not everything gets detected as well. One thing that is quite good in this example, just randomly, is that these ions, the Y and the B's here, seem to overlap, okay? They're very, very close. The masses are different, but that sort of shows how important it is to have a really good mass analyzer because we need to separate them out. If we can't, then our B and Y series basically stop and we can't reconstruct the sequence. Obviously, we have thousands and thousands and thousands of these. So it's not like we can do them by hand. That would be insane. And so basically, all we have to do is to teach the same rules to a computer and then get them to do the dirty work for us. The problem with that is that because we're dealing with such amazingly complicated ancient samples, sometimes if you have sort of a trained eye, it's always a good idea to sort of look at your product ion spectra and see whether the assignment that is done by the software is actually something you agree with, okay? Once we have the peptide, then boom, we go and search it against a database. And I'm not going to talk about this that much, but the point is that this is the starting point for unlocking, I guess, the composition of the proteome. We see which peptides we find, how specific they are. Some peptides you find everywhere, some peptides are very specific. Hopefully you get the really specific ones in your sample. And in this case, and in most experiments for the proteome of PAASTA mixta, you get a protein ID, and then you get your sequence coverage. For example, we found farfalle, but we've confirmed farfalle with the source as well, because if we don't find this peptide, it's not enough to find one peptide to call protein ID. We find tagliatelle, we find penne, we find helice or fusil. We find many different components of our proteome, and this is the basis for all the applications. Basically, this is the basic things that one needs to understand to then do, you know, shotgun proteomics on ancient samples. So really quickly, I am just going to share a few thoughts on things that you should know, you should think about, I think, from my perspective when making pasta. So pasta, you don't really have pasta on its own because that's just great. It's awful. So pasta calls for a sauce. And sauces are very different, and different sauces call for different shapes of pasta. The interaction between the shape of the pasta and the other components is important. Spaghetti call for tomato sauce. Lasagna, they really need something a little bit more meaty or chunkier in there because they're like these feta sheet looking type of pasta here. Alpha helices, fusilli, they work quite well with chunky bits of, they can accommodate quite nicely bits of veg or bits of fish or bits of meat. And so this is, again, a silly analogy, but it has to sort of, the idea is to stimulate the thought about how some protein shapes, some bits of structure of the protein facilitate interaction with other molecules, with other organic stuff that we have in the sample. We can't just forget about it because it does have an important role in, for example, how good we are at extracting proteins from our sample. So we can't ignore those. And there is a new set of AI-based tools that can actually help us start visualizing some of these interactions. And this maybe in the future will become more commonly used in the field as well. We know that the interaction between bits of the protein and the inorganic substrate is also quite important quite often. Sometimes it even allows us to have deep time preservation of some of our proteins. And again, of course, diagenesis, moving on from this, is important. It matters. We need to identify these proteins. And if we are missing many, many bits of that protein, we just cannot identify it. And the sad thing, I guess, here is that the bits that are missing quite often are the ones that are the most informative. And again, it's sometimes a question of shape. Because we have flexible bits like these bits here, these bits here in the protein. And these are the proteins, these are the bits that can actually accumulate mutations without disrupting the whole of the protein structure. And so if we think about phylogenetic implications of looking at protein sequences, these bits are the ones that actually matter. And yet these are the ones that we tend not to find. And so this is all this work from our group where Matthew and his team actually did this really clever thing of trying to substitute all the possible avian sequences in this particular region to try and figure out if our mutations were disrupting the sequence or not. And this is quite nice to see where precisely we are missing things and the effect that this can have on our applications. Data generation matters. There is, you know, a 4-hour talk on this to be had. There is also handily a guide to ancient protein studies that can sort of highlight some of the most important ones. Here, I just want to just say one quick word in support of LC method development, because we always think about having excellent mass spec sort of instruments, state of the art, and all of that. One of the best non-collagenous protein coverage I've seen is comes from the work of Carmen now in our group, together with Bruno Martinez Haya, the Universidad Pablo de Olavide in Spain, and they are using a 260-minute LC run to separate the hell out of this bone proteome. And so yeah, this is just something to keep in mind, I guess, when we think about how we generate data. Data analysis, I'm really not going to talk about this in any detail, but I find this paper really interesting, and the work that the team has been doing super interesting, because we always kind of knew that we were not very good at identifying things from ancient samples. What I really find shocking is how bad we are identifying things from modern samples as well. So there is a lot of work, and I know that you guys, a lot of you are working on improving these bioinformatics sort of approaches. And finally, I'm just going to throw this out there. Sam was talking about birds. We are working on birds a lot at the moment. We had a couple of projects. One is ongoing. Aviark and Maria Codlin has been looking at the impact of intra-species variability. And so she's got several genomes and several transcribed translated protein sequences from these genomes, and this is from the mallard, Anas platyrhynchos, and she is finding quite a bit of intraspecies differences, which is interesting, not unexpected, but this is something that, again, I don't think is being addressed systematically, and it's important for PMF-based approaches, but also for shotgun, because it kind of means that we need to have databases that are able to encompass that natural sort of biological variability. And that means a lot of work. One thing I want to say, we have an informal subgroup of the bird working group of ICAS, and it's called Parrot. And it's something we're going to put together. We are putting together Zoom's collagen and eggshell protein sequences for birds, and so we have several labs across the world that are working together to do this. So we'll send a bunch of emails out soon on this, on Parrot. Finally, something really banal, but again, sometimes it's banal, but it's important. We need to think with the head of the biomolecular scientist, but with the archaeologist sort of understanding, appreciation, and respect for context, be that a physical context the sample comes from, its biography, but also the historical context, the cultural context of the sample as well. And always thinking about where we started from, it's important. So shotgun proteomics can generate a mountain of data, but this mountain of data is only really useful in two situations: when we can make an archaeologist's dream come true, or even better, when we can inspire archaeologists to dream new dreams as well. And so on this, I'm just going to leave you with a bit of advertisement. What better venue than Torino to show us how the PAASTA community can generate new archaeological dreams. And with this, I am concluded.